Hi, class. So, in chapters 1 and 2 of the textbook, we learned about the various branches or subdivisions of biopsychology, and we were also introduced to a number of research methods and imaging techniques. So, in this presentation, we're going to be putting all that together and briefly analyze the topic of sleep from the perspectives of neuropharmacology and cognitive neuroscience. Before getting into the analysis, we need to first know what neuropharmacology and cognitive neuroscience is, as well as determine which two methods we'll be using for our hypothetical research on sleep. But we'll get to that. Let's first understand the exact definitions of these branches, their roles and jobs, and how these all relate in their own unique ways to the research of sleep. So, first, neuropharmacology is the study of how drugs affect cellular function in the nervous system. In this case, how drugs affect sleep, or perhaps thinking a little bit outside of the box, how sleep can affect the efficacy of certain drugs. Note the keyword here, cellular function, which is to serve as a hint for which methods are going to be used. Are we going to be screening this as just a picture or in real time? So, cognitive neuroscience is the study of biological structures and processes underlying cognition with a focus on neural elements of mental processes. So, in this case, how sleep or a lack of can affect your cognition. Cognition being, your thoughts, your consciousness. This could be your language, your thinking, decision making, motor skills, learning, etc. Right? And how sleep can affect that. Um, Keep in mind neural elements is the key word. We should also think about how to measure it. Perhaps there's less neurons firing in a certain area when you're tired. So, for selecting methods, given our current selections right now of sleep and our two branches, I think we can start ruling some methods out. And first, we can start with invasive and non invasive. I think in this case, non invasive is preferred. Generally speaking, if you're using an invasive technique, there's probably already something wrong with the patient, or there's something pressing that you need to investigate. And generally speaking, it's not really an ethical option um, for the study of sleep. It just doesn't make sense. Next, we have structural versus functional imaging. Structural being still image, and functional being in action or in real time, so over a period of time. And remember, we talked about how there's a focus on cellular function and neural elements. So we're going to be finding ways we can measure changes that may occur due to sleep or vice versa. I think in this case, we definitely prefer functional imaging. Our selected EEGs and the PET scan as our two methods. EEGs are very similar to MEGs, although MEGs do provide better spatial resolution at a greater cost. Ruled out TMS because it actually alters how the brain works, and because the screening portion of our research is largely observational, I don't think it'll make too much sense. EEGs work by attaching a cap with electrodes to the subject and measure the electrical activity of the brain with excellent temporal resolution to the millisecond. They're frequently used in the study of sleep, as you can measure brain waves over a span of time, as you can see in the diagram over here. The PET scan works by placing a radioactive molecule into your subject, which reacts in certain areas of the brain to emit positrons, which are detected by a scanner to form a 3D image of the brain activity. It's typically used to detect and monitor diseases, so it may not be too practical here, but we'll find some creative uses for it. Although I would have just chosen one if I could. All right, finally, how will these methods be used by people in these fields? So, first, neuropharmacology, how drugs affect cellular function. In the case of sleep, I think this one's pretty straightforward. With the EEGs, you can just measure the brainwaves. You're probably going to have a control group and also a drug affected group. The drugs don't necessarily have to be illicit drugs. They could also be, say, melatonin, for example, and kind of just comparing the brainwaves of the two groups together, seeing if there's any significant differences. So you can kind of determine a correlation between the two. We can use the brain waves and break it down into the 4 stages of sleep and determine if there's any differences in each individual stage. I think for the PET scan, for neuropharmacology, it might be a bit more difficult. I would take a more longitudinal approach in this case and determine if there's any correlation between a certain drug, a certain amount of sleep, and various diseases, so you can determine risk factors for that disease, maybe heart disease, maybe cancer. In cognitive neuroscience, EEGs would be used in a similar manner, where you can take a group of sleep deprived subjects as well as a group of subjects that sleep for 8 hours a day, for example, and assess the brain function of the two and compare it. Other uses can be to link perhaps a lack of sleep with memory loss, with other cognitive capabilities, maybe stability, walking. Just like before, the PET scan here can be used in a longitudinal manner in a correlational study to determine whether hours slept has a relationship with the development of various cognitive diseases that haven't fully been figured out yet, such as Alzheimer's, Parkinson's, MS, to better understand the risk factors involved. And after determining a strong link between any two, it would make more sense to conduct further study with other methods that can hopefully prove a cause and effect relationship. So, hopefully, now you have a brief understanding of how various research methods can be applied in different fields of biopsychology. Thank you for watching.